Hello, and thank you all for taking the time to register. And I'd like to welcome you all to our winter webinar series. This first is titled Solving Current and Future Challenges for Meat Control. And it will be presented by our very own Dr. Liberty Sabanda. Dr. Liberty holds a PhD in pharmaceutical science and a master's in international business. Sabanda has an extensive experience providing solutions to government residue monitoring programs and testing laboratories throughout Africa, Europe, and North and Latin America. I will now hand over. Okay, so I'd just like to firstly introduce myself. I'm Patrick Murphy and a marketing executive here at Randox. Just a little about Randox is we were established in 1982 and we have a consumer base in over 90 countries. Every hour over 1,000 Randox food tests are being carried out globally. Investment in research and development has increased steadily, and we have now over 30 staff dedicated to development of new assays. And I will hand you over to Liberty.
Hello. Sorry. Well, good morning to others and good afternoon to others as well. And welcome to our webinar today. Like my colleague Patrick has already, introduced Randox. So we are happy to host you today, just giving you some perspectives on current and future challenges regarding meat testing and quality control. Let me see if I have complete control on my side. I'm still trying to go to the next slide. Yeah. So the question that I usually have, or maybe some of you might actually have this question as well, is what is happening to our meat today? There are various stories going on regarding different aspects of meat, meat quality, meat, whatever is happening in the market. But one thing that I would like to, just allude to, or maybe just, give, talk about briefly is, the global meat industry, who is eating, or consuming, how much of the meat, who's winning this race. But we see that countries in Latin America are eating quite a lot, consuming quite a lot, as well as in the US and the rest of North America. If you look at Australia and, countries like China, Russia, there's quite high consumption over there. But countries in the Arab countries as well, especially in the Middle East and African countries, there's moderate consumption to low consumption. So, and you can see this here illustrated here on the graph on the right, where you have, Kenya has, very low consumption rates, as well as India. But if you look to the countries in the East, like China, in Europe, the UK, North America, Canada, and so forth. So their consumption is pretty high. And also, so that means, of course, that where there's high consumption, also it means good for the meat, good business for the meat industry. So, but where there's good business and things are booming, you know, you begin to have problems as well. We have seen, you know, headlines like this, you know, horse meat scandal in the European meat market. We have seen some headlines concerning antibiotics in poultry meat. So this is to do, you know, with you know, farmers who are culling their livestock and putting it into the food processing chain without respecting the withdrawal periods after using antibiotics on their flocks. And this has to do with minimizing costs and, you know, rushing in for profit, you know, at the expense of the health of the consumers. So we have also, you know, chicken meat rife with antibiotics. You know, this kind of headlines have been coming up, popping up in the media. Some companies, food companies, I'll refer here to McDonald's, you know, they have, you know, actually previously they had set a date, you know, to eliminate or at least reduce, you know, antibiotics in the meat that they use for their burgers and different types of their products. So which is a good move, you know, at least to see that from the food industry. But I think the biggest concern really is to do with you know, antibiotic resistance in human pathogens, you know, ones that are developing from animals into humans, which is called zoonotic pathogens. So that is where the problem is. And so therefore, even in this headline, they say testing will help restore the trust, but not only restoring the trust, restoring the proper condition which we need to live in. So talking about antimicrobial resistance and zoonosis, a lot of the problems really are coming from livestock production. So this is where the biggest problem is regarding antibiotic stewardship, you know, application, you know, and being able to observe withdrawal periods. So we have almost 70% of antibiotics in all medicinal remedies being used for livestock production, and only 30% of antibiotics are consumed by humans. If you look further, you'll see that what is about 31 of these really are used in animals as well as in humans. Therefore, if any bacterial pathogen in any disease type becomes resistant to these 31 antibiotics, humans are in trouble. So this is why there has to be control and proper stewardship of antibiotic usage. Looking at some work that has been done before, you know, from 2000 to 2018, you know, a study shows that there was an increase, especially in swine, or in pigs, let me say, that there was a 50%, more than 50% increase in antimicrobial resistance. But this particular one actually was in chicken, sorry. And in pigs, there was also a quite high increase in antibiotic resistance as well. So this actually paints a picture, you know, that in these cases where there was an increase in antibiotic resistance, that means whatever they were doing in that work, so that means that there was a failure in disease treatment. So that means whatever they were trying to do with antibiotics to treat animals or use the antibiotics for, actually treatment failed because there was antibiotic resistance. And therefore now it turns our attention to the increase in antimicrobial resistance zoonosis. So that so there is a transference now of antibiotic resistance from animals into humans. So those pathogens, bacterial pathogens, which are resistant to antimicrobials, they also now transfer their resistance into human pathogens. So that is, zoonosis actually refers to the commonality of diseases, animal diseases and humans. So whatever disease that is found in animals and is also is found in humans. So there is that transfer of disease expression. So this is zoonosis, the transfer of disease from animals to humans. So this data here at the bottom, it shows that in 2017, when they analyzed, fattening pigs, they found that there was almost a 79%, multi-drug resistant pathogens. And in 2015, again, fattening pigs, it was about 82% of multi-drug resistant pathogens. And in pig carcasses, there was 77% multi-drug resistant pathogens. So this is a big problem. So, but because of this, historically, there's always been veterinary drug regulations. And these come in the form of maximum residue limits in animal products. So what every farmer is aware of or is supposed to do, they're supposed to apply antibiotics for animal treatment and respect the withdrawal periods. So in quite a number of cases, these withdrawal periods are not observed, and this ends up with contaminated animal products being introduced into the retail market, where consumers, of course, become exposed to antibiotics and any other, resistant factor that could be present in those products. So therefore, different countries, different regions have come up with maximum residue limits. These are what we call, food contaminant regulations. Almost all of these are really underpinned by the Codex Alimentarius. So Codex is global. This is the global sort of regulations on which every country really bases its regulations upon. But we have, regions like, the European Union. The European Union has, I think, the most stringent regulations covering the wider scope of residues or analytes. And then you also have the one from the USDA, FDA regulations as well. So all this is done in the effort to reduce antimicrobial resistance, and therefore, trying to protect any future pandemics where, our drugs might fail. So there is what you call the national residue monitoring programs that are put up by almost every country, but unfortunately, not really every country does that. So just to give you a brief overview of countries that are doing this, this is not very much up to date, maybe legs behind by something like five years or so. There could be updates in some of these countries, but you'll see that all these countries here, a lot of them, they have a lot of gaps for things that, different types of animal products that are not being monitored, tested for and controlled within their borders. And so that means the local consumers, that population, they could be exposed to a lot of antibiotic residues because nothing really is going on in a lot of these countries. But what is common in most of these countries really is aquaculture. Aquaculture has, it seems to have a lot of attention from most of these countries. So as you can see, aquaculture is almost very consistent in terms of application of national residue limits. So, So unfortunately for a lot of them, there are quite a lot of gaps, and some of them are actually doing nothing in terms of national residue control. So now, unfortunately, the inability to use or and implement national residue monitoring programs, you know, it affects international trade, because if you are producing any type of food, you are active in the international market, and that's where, you know, you get some of your economic performance, you know, for the country. But if you do not have a national residue monitoring program, in most instances, you will not be able to access international markets. Therefore, regulations play a very significant role in international trade. So if you look here, this is all to do with veterinary drug residues in animal products. The EU, the US, and Codex. You know, Codex, like I said before, is quite international, and but Codex misses quite a lot, as you can see. They miss if you're basing your food quality on Codex regulations and standards, you're going to miss a lot. And when it comes to international trade, whatever you are trying to trade might be found to be lacking in terms of assurance in other countries or regions like the US or the European Union. So you won't be able to access those markets, because look at the EU, there are regulations for all these antibiotic residues, as you can see. On the other hand, the US as well misses quite a bit. So they are regulating less than the EU. Okay. So so here as well, if you do not have national residue monitoring programs, so it means that or if you have less, it means that also the methods that you are using for testing are going to be in line with your, you know, your national residue monitoring program. I've seen that in countries where they have quite an extensive menu and robust national residue monitoring programs, they have instruments like the LC-MS, but this is quite an expensive setup. But a lot has been, even historically, a lot of labs and jurisdictions have been using the bacterial inhibition test. So in the bacterial inhibition test, you have bacteria in this agar media. There is bacteria, this is Bacillus stereothermophilus, so which is quite sensitive to, quite sensitive to beta-lactams and moderately sensitive to other antibiotics. So what happens is that if you put a sample in this media, a meat sample, and if it contains antibiotics, so this bacteria is, if it is sensitive enough to that antibiotic or level of antibiotic, it will not grow. Therefore, the media will stay blue. So this is a blue indicator, which is integral in the media. So it will stay blue. So that will tell you that after incubating this test at 37 degrees, if it stays blue, therefore the sample is positive. However, if the sample does not contain any antibiotics, it will turn yellow because the bacteria will begin to grow, produce acids, and that acid is going to turn the indicator yellow, from blue to yellow, showing you that the bacteria is alive and the sample does not contain any antibiotics. Therefore, it is a good test, broad spectrum. You can test quite a number of antibiotics in this test. However, the problem is that it misses quite a lot. So if you look at the beta-lactams, it misses at least 50% of the beta-lactams. And then tetracyclines although it is sensitive to the tetracyclines, it misses the LODs required, to attain the MRL. So the LOD for tetracyclines is higher than the MRLs. So, and then again, for sulfonamides, it is sensitive to only a few sulfonamides. More than 90% of the sulfonamides it will miss. The aminoglycosides, 100% LOD is higher than MRL. And then look again at the macrolides. Again, the macrolides is not sensitive enough to MRL. The quinolones, on the other hand, there is absolutely no sensitivity to quinolones. So you miss quinolones altogether. So that means if you use this test to test your exports or whatever, so that means you are going to end up with antibiotics, in your food, whatever the consumers are going to have. They will have antibiotics in their food. So now, the other tests, now just looking at chromatography, there is of course ELISA in the meantime, but I will just concentrate on these because they are used quite a lot as well in some residue monitoring programs and for they are used for export, to verify the quality of exported goods. So for HPLC, it is a high cost, high running cost kind of setup, because you need immunoaffinity columns to clean up, because you have to achieve very clean baseline in your chromatograms. Another problem is that, for every analyte that you have to test or group of analytes that you have to test, you need a specific method. And that specific method will take you at least a day and a half to complete. Okay, so including sample cleanup and then total run. Another thing is that you need standards, for your calibration curve, and all these things here are quite expensive. Okay, your sample prep, your standards, and so forth. So it's quite expensive to run. On the other hand, the LC-MS/MS, you can do quite a lot. Or you can test quite a lot of analytes. You've heard of multi-residue method, LC-MS/MS method, which is very, very powerful. And the sensitivities here are really, really good. You do not have to worry regarding, being able to achieve the sensitivity required to verify MRL adherence. And then, but the problem is that it is very, very expensive. Most labs, they cannot afford. So very high initial investment, but the other thing is you need well-trained personnel. And LC-MS/MS personnel, they're very, very hard to keep. Once you've trained somebody, very, very hard to keep. So, and sample cleanup costs are very high as well. And the standards, like I've mentioned for HPLC, for the calibration curve are very expensive. But in addition to that, LC-MS/MS requires what is called an internal standard. And this is quite expensive. Usually it is in the form of an isotope, the isotope of a standard, whatever standard you might be using. And then you look for an isotope type variation of that standard. And it is very, very expensive. So, and turnaround times as well. So very, very long. So these are the issues that we have in terms of meat testing and, trade and being able to achieve to the proper regulations in the different jurisdictions where we're trying to do business. But if I may just give you a brief snapshot here of what you call the RASFF Portal. So this is the EU rapid alert system, which monitors every consignments that come into the EU area, and they are tested at borders. And you will see that there's been quite a lot of rejections. Say here at the bottom, you see China 3 times, chloramphenicol with nitrofurans, So this is border rejection. And then we have Argentina here, azapiron, rejected by Switzerland. And then we have Brazil, you see we have diclofenac. The US, we have ractopamine. So this is a prohibited substance. Now, the problem with ractopamine is that it is not prohibited in the US. So now, to trade with the EU, when you are using your local standards, and then there is now a conflict because now this is actually prohibited in the EU. So you also have there Italy, Brazil again, border rejection for like the tetracycline here, doramectin. One of the ivermectins, doramectin, and you also have from Italy, of course, sulfamethoxin. So the tests, wherever you go, you find that there are tests. So, but how do we breach that? So this is what we are trying to come up with kind of solutions today. How can we breach these issues? I mean, we have varying regulations for Codex, which is worldwide, and then for the US and for the EU. So how can we actually help people who want to trade across these borders? And this is why we talk about the Biochip array technology. So we actually start with the food industry itself to say, you can cover quite a lot of substances within your food manufacturing facility without even going to the labs, with a very rapid turnaround time and multi-residue analysis platform. And this is quite an effective platform that you can use to ascertain your production within your facility. You can do all the antimicrobials, your quinolones, your tetracyclines, your aminoglycosides, macrolides, your phenycols, your beta-lactams, sulfonamides, nitrofurans, anthelmintics, all the way to your steroids. But in addition, we do also pathogens. And we also include COVID-19 in there. So we have a wide range of applications just on one station, and this actually fits very, very easily because it is simple. It fits very easily in a food manufacturing or processing environment. So it actually empowers the food processor to be able to achieve quick batch releases. So there is no need to send samples to the lab immediately. So you can actually release your batches pretty quick. If ever you have, it might happen that you have any positive samples, when using the Biochip array technology, then those positive ones, you can send them to the lab, for confirmation. So confirmation here, according to the regulation, confirmation means the identification of, positive identification of the contaminating substance. Okay, because the concentration determined by the Biochip array technology is very, very accurate. It compares very well to the LC-MS. So that means you also reduce your turnaround times. You also reduce the time you have to hold your production. It helps a lot, for manufacturing environments. And yet, it also gives a lot of throughput as well to the routine analysis lab. So here, what you have actually is a very high throughput system. The evidence investigator using the Biochip array technology that can come in and marry your LC-MS analysis, and you're able to do a lot of samples. 1, 2, you're able to do a wide range of analytes within 2.5 hours. Okay, and then any positive samples, then you can send them to the LC-MSMS. So, I know that, people talk about doing screening on your LC-MSMS, which is great, but it actually allows you not to overload your LC-MSMS. You have to protect your source, and you have to protect your rods in there, and make sure that you are not running down your system. And also it helps you as well to reduce your analysis cost as well, and your turnaround time, because within 2.5 hours, you can imagine you can do more than 45 samples within 2 hours for myriads of analytes. And so just talking about the Biochip array technology itself, this is a platform where you can do multiple samples and actually generate multiple results. And one biochip carrier contains 9 biochips. And one biochip well is used for one sample. And within that one sample, you can do quite a lot of analytes. And just for QC purposes, there are 3 spots. There's a reference spot, a control spot, and another correction spot. So 3 of them to make sure that the orientation of the biochip is correct and the identification of each analyte is optimum. So the benefits of the biochip actually is simple sample preparation. It's very, 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 very easy. If you look at the technology, this is a multiplex technology. But if we are to refer, actually, like, what are we talking about when we say the method is simple? So you take 1 gram of tissue sample and add 19 mils of wash buffer. So this wash buffer is provided in the kit as well. And this is vortex for 10 seconds and then centrifuge for 10 minutes at 4,000 rounds per minute. So the supernatant then is diluted. So that's it for sample prep. No additional costs or anything like that. And within 20 minutes, your sample is ready for analysis. And then what you do then is to add about 150 microliters wash buffer of acid buffer to the biochips, and then add your standards, which are also provided in the kit. No additional cost to this. And then your diluted sample as well, and incubate for 30 minutes. And then after 30 minutes, then you add your multi-analyte conjugate and incubate for 1 hour. And then wash the biochips, and then add the substrate there to generate the chemiluminescent result, which is you use luminol and peroxide. So and you have multiple calibrations that are generated for each specific analyte, and this then allows the system to do the calculation of the concentrations of each specific unknown analyte in each sample. Okay, so just to talk briefly about some validations that we've had. So this was done in Fougères in France, and it was done by ANSYS. I don't know, some of you might be aware of this world-class lab in France. And they did actually the validation of the multi-residue or antimicrobial residue test based on the biochip. And this test can actually do 6 groups of antibiotics: your quinolones, your beta-lactams, your phenicals, aminoglycosides, macrolides, and tetracyclines. And it can detect these quantitatively. And so what they did was then to look at the CC beta in comparison to the MRL. So this MRL here is the EU MRL. And you will see that from these results, the CC beta was always lower than the MRL, the EU MRL. So that means if you use this system to test all these antibiotics, you can test them quantitatively. That is one. And also you can test them, whatever result you get, it will tell you whether your consignment or that specific sample representing whatever consignment does conform to EU MRLs or USA MRLs or Codex. So because we are so sensitive, we are sensitive at a lower level than the MRL for the EU. So in addition, just to show a little bit more regarding other tests that are there. So we cover quite a lot of antimicrobial tests, the ivermectins as well, anthelmintics, the coccidiostats. So very, very robust menu of everything that is regulated out there. In addition, we also have ELISA for those that are testing individual methods, of course. So we still cater for the individual ones, and we have quite a wide choice, you know, of antibiotic, also growth promoters as well, non-steroidal hormones and stuff like that. Let me just also mention that we are not forgetting those that are using HPLC and LC-MSMS. We provide you know, immunophenetic columns for growth promoters, for steel beans as well. And you can use these tests so many times. You know, you just do a column regeneration, and you can repeat over and over and over again the usage of these tests. So so you can use them for HPLC sample cleanup. You can use them for LC-MSMS sample cleanup as well. So I would like to thank you for your attention today, and we'll give you back to, we'll give you back to looking for.
Okay, thank you very much, Liberty. I'm sure there's a lot of information there that everyone can take from that.
Yeah.
We will be doing a quick Q&A for any of the participants that have any questions for you.
Okay.
So if anyone has any questions, to fire them through, and we'll ask one or two towards you. There is a dialog box with a Q&A. So if you have any questions, make sure you type into said box.
There is a hand there.
Yeah, there is a question here from Michael. He's asking, could you please revisit the benefits of biochip array technology?
Okay. So so basically, so the first one is that you are able to test many analytes in one run. Okay, so for antibiotics, for example, you are able to test your quinolones, your beta-lactams, aminoglycosides, your phenicols, tetracyclines, and macrolides all in one run. And this is quantitatively. So, so the other things are like ivermectins and all the anthelmintics. You can run them all in one run quantitatively. So these are just examples. And then the sample preparation is very simple. So there is no sample cleanup, okay? And it's very brief. It's maximum 20 minutes. It's only shaking and centrifugation. And then you dilute your sample. So that is 2. So another thing is that you do not have to, you do not need a big lab space, and there's not much training that is required for you to be able to be proficient with the biochip array technology. You know, if you have used ELISA before, it does help. Okay. The other thing is that the running cost is very, very low. The running cost, we compete with, in terms of result with ELISA, we compete a lot with ELISA, okay? So those are some of the benefits you have with the biochip array technology.
Okay, perfect. Another question here is, what meats are validated on your platform?
Sorry?
What meats are validated on your platform?
We actually validated muscle, kidney, liver. So if you talk about kidney and liver, these are very difficult matrices. So those are actually validated, so which is great. And I'm just trying to think of any additional, but those are the main ones when you talk about target organs, which are usually referred to in MRL or in maximum residue regulations. So those are the main ones. So, but we also have, I think, in serum as well, I think, yeah.
Okay, thank you very much. And another one here is, a consumer country like Qatar, how is this instrument positioned in central food labs?
How is it, what, sorry?
Positioned in central food labs?
Yeah, well, I think it is the best place as well, because this, you can actually place it to complement any higher end instrument you might have, because what you do not want to do is to load a lot of your sample volumes onto the higher end instruments. For example, if you have an LC-MS, therefore already your the LODs in the sensitivity that you find on our biochip array technology is very, very competitive enough and very low enough to achieve to lower than the MRLs of the EU. So that means whatever you have, you can screen for exports, for imports. So this actually equips the central lab, for example, in Qatar, to be very, very effective and achieve very short turnaround times with robust results. So it's a very optimum place to have the biochip array technology, especially the evidence investigator in a situation like that.
Okay, thank you very much, Liberty. For anyone that didn't get their question answered, we will be emailing you personally to go through any questions you have. And I'd just like to thank you all for taking the time to register. And again, thank you to Dr. Liberty for his wonderful presentation.
So thank you, everyone.
Okay, thank you.